Imagine a tumor so resilient that starving it of oxygen and food actually makes it stronger.
Yeah, it sounds like science fiction, but it's very real.
Right. I mean, for decades, treating brain cancer has felt a bit like fighting a ghost. You use every weapon in the modern medical arsenal, surgery, radiation, chemotherapy, and just when you think you've cornered it, it changes its shape, changes its diet, and comes back more aggressive than before.
It's incredibly frustrating for oncologists.
Absolutely. So, welcome to this custom-tailored deep dive. If you're listening to this, we know you are a learner, you crave those aha moments, and you want the complete breakdown of complex mechanisms without feeling overwhelmed.
You're not here for the surface-level summary.
Exactly. We are taking a massive stack of research today, specifically a highly detailed paper from Nature Cell Biology. And our mission is to completely deconstruct it for you. We're going to go through this source material thoroughly, unpacking, every single concept, mechanism, and detail.
We won't gloss over the hard stuff.
No way. We'll use stories and analogies to make it stick.
It is a phenomenal piece of literature to dig into. We're looking specifically at glioblastoma, or GBM, and more specifically, the IDH wild type.
Which is pretty much the worst of the worst, right?
Unfortunately, yeah. It's one of the most aggressive, adaptable, and frankly, incurable brain cancers in existence. Even with all the data we've gathered from the modern omics era, the 5-year survival rate remains a devastatingly low 5 to 10%.
Okay, let's unpack this. Why is GBM so incredibly hard to defeat? Like, what is it about the architecture of this specific enemy that makes our most advanced therapies fail so consistently?
Well, the defining characteristic we have to understand first is tumor heterogeneity.
Heterogeneity, so meaning it's not all the same.
Right. You aren't fighting a single uniform mass of identical rogue cells. You're fighting a constantly shifting, diverse coalition. The source paper highlights a profound cellular diversity within GBM, and they basically categorize the battlefield into two main cellular players.
Okay, what's the first one?
First, you have the glioma stem cells, or GSCs.
GSCs. So these would be like the roots of the tumor, right?
Yeah.
The foundation.
Exactly the right way to visualize it. GSCs possess self-renewal properties. They can endlessly copy themselves and propagate the tumor.
Like weeds in a garden.
Yeah, but their most dangerous trait is that they harbor intrinsic therapy resistance mechanisms.
Wow.
They are the primary drivers of tumor progression. I mean, if the tumor survives a round of chemotherapy, it's usually because the GSCs hunkered down and weathered the storm.
Right, because the roots survived, and the second group of cells.
Those are the differentiated descendants, or GDCs.
Okay, so if the GSCs are the roots, the GDCs are the sprawling branches and leaves.
Precisely. They adopt highly specialized phenotypes. These are the cells that substantially contribute to the physical bulk of the tumor burden.
They make up the actual mass.
Yes, and they drive the invasive spread, literally pushing their way through healthy brain tissue.
But the environment they're invading isn't exactly hospitable. The paper paints a picture of the tumor microenvironment that is fluctuating and incredibly harsh.
Oh, it's brutal.
We're talking about extreme hypoxia, right? Dead zones with almost zero oxygen. Plus, there's a severe lack of nutrient and glucose delivery.
Right, because the blood vessels that the tumor hastily builds to feed itself are completely disorganized. They're poorly formed and leaky.
So it's a highly stressful, deeply toxic microenvironment. For a normal, healthy cell, finding itself in that environment is just an immediate death sentence.
100%.
So if I compare this tumor to a specialized army trying to survive in an arid, hostile desert.
I like that analogy.
If they don't have standard supply lines for food and fuel, they must be fundamentally mutating their own internal engines to survive on whatever alternative fuel they can scavenge, right?
That brings us to the core concept of the paper, which is metabolic flexibility or plasticity.
Plasticity, meaning that they can mold themselves.
Exactly. The tumor cells are forced to constantly adapt their metabolic requirements just to survive in those hypoxic dead zones. They completely rewire how they consume and process energy.
That's terrifying, but also kind of amazing.
It is, and because of this, researchers have realized that the tumor cells are prime targets for therapies that go after metabolic vulnerabilities.
Okay, let's just do a quick consolidation checkpoint here so we don't lose anyone.
Good idea.
So to summarize what we just covered, GBM is deadly and heavily diverse, mainly split between the root-like GSCs and the sprawling GDCs.
Right.
And it survives a completely toxic, starving environment through metabolic flexibility.
Perfect summary.
So for you listening, Think about a time you had to adapt to a severe lack of resources, like maybe your car broke down without your phone and you have to find your way home. How did your strategy change? Keep that kind of desperation in mind as we look at how this cancer adapts.
That desperation is key.
Because when you're looking at a cancer that survives a harsh desert of a microenvironment, you have to realize it's completely rewritten its own survival rulebook.
It has.
It forces us to ask, how exactly is it pulling off this metabolic magic trick? How is a cell functioning without oxygen or glucose?
To answer that, we have to zoom way, way in.
Down to the microscopic level.
Past the tissue, past the cell membrane, down to the subcellular level, we have to examine a structure called MAMs.
MAMs, mitochondria-associated membranes. Let me build a visual memory aid for you listening.
Go for it.
Picture two massive, distinct factories inside the cell. The endoplasmic reticulum, the ER, is the manufacturing plant. It synthesizes proteins and stores raw materials.
Right.
And the mitochondria is the power plant, generating the actual energy the cell needs to run.
Yes.
So MAMs are essentially temporary, highly regulated suspension bridges built between these two factories so they can quickly trade supplies internally without having to go outside into that harsh, starving environment.
That captures the dynamic perfectly. A MAM is not a separate, free-floating organelle.
It's not its own little factory.
No, it's a physical tethering point, a dynamic crosstalk contact site between the ER and the mitochondria.
Okay.
And the researchers in this study wanted to know exactly what these bridges are made of in GBM cells. They took patient-derived GBM cells across 3 distinct molecular subtypes:
Which were?
classical, proneural, and mesenchymal.
But wait, isolating a temporary microscopic bridge inside an already microscopic cell sounds nearly impossible. Why couldn't they just use standard cell extraction methods?
Because standard extraction would just rip these delicate contact sites apart. It's too violent. Instead, they used a highly specialized technique called Percol fractionation.
Let's break that down. Percol is a type of silica colloidal gel, right?
Yes, exactly. They carefully burst the cells open, place the contents into a tube filled with this specialized Percol gradient, and then spin it in an ultracentrifuge at massive speeds.
Wow.
Because the gel has different densities at different levels, the heavy cellular components sink to the bottom, the light components float, and the intact MAM bridges settle into a very specific extractable layer right in the middle.
That is brilliant. Just separating them by weight, basically.
Pretty much.
And once they isolated these bridges, what did they find?
They ran comparative proteomics.
Using mass spectrometry.
Yes, to identify every single protein present in that layer. And they discovered that major MAM proteins were clustering in the exact same protein-protein interaction network across the aggressive GBM subtypes.
What kind of proteins are we talking about?
We're talking about a dense network of structural and functional proteins, things like VDA C1, 2, and 3, IP3R2 and 3, and a chaperone protein called GRP75.
Whoa, okay, alphabet soup there. We definitely don't need to memorize all of those acronyms, but what do these proteins actually do? Like, are they the bricks of the bridge, or are they the traffic cops directing the supplies?
Well, they're kind of a bit of both. The VDAs and IP3Rs form the actual physical channels, like the scaffolding and the roads of the bridge.
Okay, got it.
And the chaperone proteins, like GRP75, help stabilize those structures and guide the molecules across.
Makes sense.
What's fascinating here is what happened when the researchers pharmacologically attacked those specific structural proteins. They used a drug called MKT-077.
Which does what?
It specifically targets and inhibits that GRP75 chaperone protein. When that protein was knocked down, the viability of the cancer cells absolutely plummeted.
Oh, wow. So if you bomb the cecal bridges, the factory completely shuts down, even if the factory itself wasn't directly hit.
Precisely.
Let's do another consolidation checkpoint right here.
Sounds good.
So metabolic flexibility completely relies on MAMs. And MAMs are these physical communication bridges between the ER and mitochondria. And disrupting them seriously hurts the tumor.
You nailed it.
So listener, ask yourself this. If you were a general trying to stop an enemy factory, would you bomb the heavily fortified factory itself or the secret, fragile bridges supplying its power?
It's much easier to take out the bridge.
Right. So that proves the tumor's metabolic flexibility is heavily reliant on these MAM contact sites. But out of all the proteins on that bridge, the proteomics data singled out one specific protein as the ultimate mastermind.
It did. The protein is called ER1.
ER1. Why did that one stand out from the rest of the pack?
Well, first, the clinical data. High ER1 expression in patients is significantly associated with really dismal survival rates.
That's a bad sign.
Yeah. Furthermore, when analyzing the 3 subtypes of GBM, ER1 was dramatically enriched in the mesenchymal subtype.
And the mesenchymal subtype is notoriously the most aggressive form.
Exactly.
It's often found clustered in paranecrotic areas, which are, the literal dead zones of the tumor that completely starved of blood flow and oxygen.
It thrives where the cell is under maximum stress. ER1 normally plays a role in protein folding inside the ER, but in these highly stressed glioblastoma cells, it gets co-opted.
Co-opted how?
It acts as a critical regulator of the MAM structure itself. Its presence physically tightens the gap, pulling the ER and the mitochondria much closer together.
Wait, so it literally shortens the suspension bridge to make the transfer faster?
Yes, it brings them physically closer. And by doing that, ER1 creates what researchers call calcium hotspots.
Calcium hotspots.
Right. The endoplasmic reticulum is the cell's main storage vault for calcium. When the MAM gap narrows, a highly efficient concentrated stream of calcium ions, Ca2 plus Neson, flows directly from the ER into the mitochondria.
Wait, calcium. I usually think of calcium as the building block for bones or teeth, not as a fuel source for a cellular power plant. What does the mitochondria want with calcium?
Yeah, it's counterintuitive, but in this context, calcium isn't a building block at all. It's a potent signaling molecule.
Okay.
Inside the mitochondria, that flood of calcium acts as an ignition spark. It binds to and directly stimulates mitochondrial dehydrogenases. Most importantly, an enzyme called pyruvate dehydrogenase, or PDH.
And sparking PDH triggers the next phase of energy production.
Exactly. Stimulating PDH supercharges the TCA cycle, the tricarboxylic acid cycle.
Right.
By driving the TCA cycle into overdrive, it drastically boosts mitochondrial oxidative phosphorylation.
Which is basically energy production.
Yes. The researchers measured this as an increased oxygen consumption rate, or OCR. Concurrently, it also boosts glycolysis, measured as the extracellular acidification rate, or ECAR.
Okay, hold on. How do you even measure a microscopic cell's oxygen consumption in real time? Like, how is that physically possible?
It's amazing technology. They utilize Seahorse XFEN96 technology, which is an incredibly sensitive metabolic analyzer.
Okay.
It physically measures the drop in oxygen and the increase in acidity in the fluid immediately surrounding the cell. So it gives a real-time readout of how fast the cells are basically breathing and metabolizing.
That is wild. Let me summarize that visually because the mechanics here are just crazy. ER1 is essentially grabbing the power plant, physically pulling it tightly against the manufacturing factory, and pumping high-octane liquid, the calcium, directly into the furnace.
Yes.
So this allows the engines to run on absolute overdrive, keeping the cancer alive even when its standard food source, like glucose, is completely missing from the environment.
That is the precise molecular definition of the tumor's metabolic flexibility. It is surviving by tightening its internal supply chain.
Time for checkpoint number 3.
Let's do it.
So, ER1 is highly elevated in aggressive starved tumors. It tightly clamps the ER mitochondria bridge together, and it pumps calcium to turbocharge energy production.
Spot on.
So for you listening, can you picture that calcium spark turbocharging the mitochondria? Let's see what happens when we try to put out that spark. Because if ER1 is pulling these factories together to survive starvation, my immediate thought as a researcher would be, what happens if we just break the bridge?
That's exactly where the researchers went next.
How do we actually target and dismantle this mastermind protein?
They proved this vulnerability using a two-pronged approach, pharmacological and genetic sabotage. Let's look at the pharmacology first. They utilized a targeted small molecule inhibitor called EN460.
Okay, EN460. How does it work?
Well, EN460 binds directly into a very specific pocket on the ER1 protein. It's called the FAD, or flavin adenine dinucleotide binding pocket.
A pocket on the protein. Got it.
Right. By occupying this space, it prevents ER1 from functioning and reoxidizing. But the really critical detail is how it binds. EN460 requires a covalent interaction with a specific amino acid site on the protein cysteine 397.
You mentioned it forms a covalent bond at that cysteine 397 site. Why does that specific bond matter so much? Like, is it just temporarily blocking it, or is it permanently jamming the gears?
Oh, a covalent bond means the drug actually shares electrons with the target.
Oh, wow.
It is a highly stable, virtually permanent lock. Once EN460 binds to cysteine 397, that ER1 protein is permanently decommissioned.
It's done.
Done, and the results were immediate. When they placed these patient-derived cancer cells under severe glucose deprivation, mimicking that harsh brain tumor environment, and treated them with EN460.
What happened?
The cell's viability, their ability to proliferate, and their capacity to migrate all plummeted.
The engine stalled out completely because the calcium spark was extinguished. But, in scientific research, drugs can sometimes have off-target effects. They might be hitting other proteins by accident. How do they prove it was definitively the silencing of ER1 that killed the tumor and not just some random side effect?
That's where the second prong comes in. They engineered a genetic attack to map the protein's exact vulnerabilities.
How?
They used lentiviral shRNA, that's short hairpin RNA. They used it to completely silence and knock down the cell's natural expression of ER1.
So they just turned off the gene entirely.
Right. But then they performed what we call rescue experiments.
Meaning they put ER1 back into the cells to see if the cancer would recover.
Yes, but they didn't just put normal ER1 back. They engineered meticulously crafted mutant versions of the protein with specific point mutations.
Oh, I see.
They wanted to know exactly which parts of the protein machinery were vital for the MAM bridge.
This is where we get the molecular blueprint.
Exactly. They mutated the regulatory cysteine pair, which is located at amino acids C94 and C99. They mutated the catalytic cysteine pair at C394 and C397. That's the actual active site.
Right.
And finally, they mutated two phosphorylation sites, serine 143 and serine 145.
It's incredible to think about the scale here. I mean, we were talking about tens of thousands of molecules inside a single microscopic cell. And by swapping out just a couple of specific amino acids, they structurally altered the entire architecture of the cell.
They fundamentally changed its physical shape. When they introduced these mutated versions of ER1, the researchers utilized electron microscopy to visually inspect the cells. They could physically see that the MAMs had structurally failed.
Wait, really? Looking through an electron microscope and actually seeing the gap widen?
Yes.
That is profound. They proved the bridge isn't just some abstract communication line. It's a physical, mechanical tether.
Complete.
By altering a single cysteine or serine, they literally push the power plant away from the factory. The distance between the ER and the mitochondria increased, the calcium transfer dropped, and the cell's energy production just crashed.
It confirmed, without a doubt, that the enzymatic and regulatory activity of ER1 is directly, mechanically responsible for tethering those organelles together.
Okay, here's where it gets really interesting, and let's do our fourth checkpoint.
Okay.
We have the molecular blueprint of the enemy's engine. We know the EN460 drug blocks ER1. We know specific cysteine and serine sites are critical. And mutating them physically breaks the MAM bridge, cutting off the tumor's energy.
That's the core of it.
So listener, we have the blueprint. The next logical step is, does this actually work in a living brain?
Yeah.
Because moving from isolated cells in a plastic petri dish to a living, breathing system is where most cancer drugs go to die, right?
It's the ultimate bottleneck, yeah. But the transition to in vivo models here was rigorous. They escalated the complexity of their models step by step. They began with human brain organoids.
Organoids, those are the miniature 3D brain-like structures grown from human stem cells in a lab, right?
Correct. They created a co-culture. So they took the aggressive mesenchymal glioma stem cells and introduced them to the healthy organoids.
Okay.
When they applied the drug EN460, it significantly halted the cancer cell's ability to invade the organoid tissue.
Wait, if this EN460 drug destroys the energy bridges in the cancer cells, why wouldn't it also destroy the MAM bridges in the healthy brain tissue right next to it? How is it selective?
That's the beauty of it, because it targets a dependency. Healthy brain cells are not trapped in a hypoxic, starving environment.
Oh, right.
They're not relying on ER01 to artificially tighten their MAMs and run their mitochondria on massive overdrive. EN460 exploits the cancer's desperation.
So it pulls the rug out from under the tumor, while the healthy cells, which are just functioning normally, are largely spared.
Exactly. That selectivity is the holy grail of oncology.
Wow. Where did they test it next?
Zebrafish larvae. Zebrafish are fantastic models because they're transparent, which allows researchers to observe tumor progression in real time.
Oh, that's wild. You can literally see the tumor growing inside the fish.
Yeah. And when they implanted cancer cells where EO1 had been genetically knocked down, the overall tumor volume in the fish was drastically reduced compared to the control group.
But to truly validate a human therapeutic, you eventually need mammalian models.
Yes. So they utilized two distinct mouse models. First, they used immunodeficient nude mice injected with human patient-derived stem cells, specifically the G518 line.
Okay.
Second, they used immunocompetent mice injected with a syngeneic mouse glioblastoma model known as SB28.
Why go through the trouble of using two completely different mouse models? That seems like a lot of extra work.
It is, but you need it to ensure the results are robust across different biological landscapes. They needed to know if a functioning immune system would skew the drug's efficacy.
Oh, I see.
Additionally, the SB28 cells actually express a lower baseline level of ER1A, meaning they were slightly less sensitive to the drug when tested in isolation. So they needed to see if the drug could still perform under less than ideal conditions.
And what was the verdict on survival? Like, did breaking the MAM bridges actually extend the mice's lives?
The survival data was unambiguous. Systemic inhibition of ER1A using EN460 provided a highly significant, therapeutically meaningful survival benefit.
That's incredible.
The control mice, who received a placebo, quickly succumbed to highly aggressive invasive tumors. But the mice treated with the inhibitor lived significantly longer.
Right.
And when pathologists examined the brain tissue afterward, they found that the highest concentration of ER1A expression was located right in the dense core of the tumor, distinctly separate from the surrounding healthy brain tissue.
That completely validates the mechanism.
Yeah.
It proves that ER1 isn't just an artifact of growing cells in a plastic dish under artificial conditions. It is a structural linchpin for the tumor's survival in a living organism.
If we connect this to the bigger picture, it demonstrates that the ER mitochondria contact sites represent a context-dependent druggable vulnerability.
Okay, final consolidation checkpoint.
Go ahead.
So we tested in organoids, fish, and mice. Blocking ER1 consistently shrinks tumors and extends survival. And it selectively hurts the cancer while sparing normal tissue.
Exactly.
So listener, seeing a concept move from a single protein mutation all the way to extending life in an animal model, what does that tell you about the power of targeted medicine? Which brings us to the ultimate synthesis of this deep dive. We started out looking at glioblastoma, a cancer that routinely outsmarts our best treatments through sheer heterogeneity and incredible metabolic flexibility.
Right.
We always thought of its adaptability as an unbreakable shield.
But this research revealed that its greatest strength relies entirely on a microscopic physical tether, the MAMs, and specifically the master regulator tightening that tether, ER1.
In a world where cancer constantly mutates its genetics to evade traditional chemotherapy, targeting the physical structure of its internal energy supply chain, literally cutting the suspension bridge between the factory and the power plant, represents a totally new flank of attack.
It is a massive paradigm shift. We're moving beyond simply trying to poison a dividing cell. We're now targeting organelle dynamics, spatial architecture, and calcium fluxes.
It's architectural warfare.
Yeah, we have found a way to exploit the tumor's own stress response against it.
They took the invisible engine of glioblastoma, mapped every single bolt, and showed us exactly where to throw the wrench. And as we wrap up, I want to leave you with a final thought to mull over.
It's a really profound implication.
We've spent this entire deep dive seeing how ER1 builds a bridge to help a highly stressed cancer cell survive a toxic environment. But think about this. If these MAM bridges are fundamental to how human cells handle extreme stress and manage energy, could this exact same mechanism be at play in other devastating diseases?
Diseases where we actually want the cells to survive the stress.
Exactly.
Yeah.
Like the neurons degrading in Alzheimer's, or the tissue dying during a heart attack. If we can chemically destroy the bridge to kill a cancer cell, could we eventually learn to fortify that bridge to save a healthy one?
It's a fascinating possibility.
The same mechanism that makes cancer deadly might hold the key to treating neurodegeneration. Something for you to ponder long after this deep dive ends. Thank you for taking this incredible high-density journey with us today. Keep asking the hard questions, and we'll see you on the next deep dive.